Back in episode 2, we drew the one-frame contrast at the heart of this. Screening searches an existing haystack for the needle, while de novo design builds the needle from scratch. This episode is about how that building actually works, the generative chemistry stage that produced rentosertib's molecule. It's one of the places where AI isn't just speeding up an old method, it's doing something the old methods fundamentally couldn't. So let's be precise about the difference. Virtual screening, which we'll cover in episode 10, takes a fixed library of already known compounds and ranks them. It's bounded by whatever someone has already synthesized. De novo generative design does something else entirely. It produces novel molecular structures that may never have existed before, optimized against target binding and drug-like properties at the same time. It's unbounded by existing libraries, and that's both the point and the risk. The point, because you can reach molecules no catalog contains. The risk, because you can just as easily generate something no chemist can actually make. How do these models actually work? Two dominant families, both borrowed from the same lineage as image and language generation. The first is diffusion models. They start from pure noise and iteratively denoise it into a valid molecular structure, often directly in 3 dimensions, positioning atoms right inside the target's binding pocket. It's the exact same mechanism as image generation diffusion, just applied to atoms instead of pixels. The second family is transformer, or language model, approaches. These treat a molecule as a sequence of text, using a notation called smiles, which writes a chemical structure as a string, and generate it token by token, the way a language model writes a sentence. The crucial difference from generic generation, the goal isn't a plausible-looking molecule, it's a molecule that satisfies several hard constraints all at once. And that is the whole difficulty. A usable drug has to satisfy several constraints simultaneously, and improving one usually degrades another. It needs binding affinity for the target, from the structure and affinity prediction in episode 7. It needs selectivity, bind the target and not its lookalike proteins, or you get off-target toxicity. It needs good ADMET properties, absorption, distribution, metabolism, excretion, and toxicity, because a perfect binder the liver destroys in minutes is useless. And it has to be synthesizable, meaning a chemist can actually make it. So generative design is really multi-objective optimization, navigating an astronomically large chemical space. Estimates of drug-like molecules run to 10 to the 60th, toward the tiny region that satisfies every constraint at once. That's exactly why brute force screening can't get there and generation can. There are 2 ways to steer that generation, and which one you use depends on how good your structural information is. Structure-based design works directly into the 3-dimensional binding pocket predicted back in episode 7, it's the most powerful approach, but it needs a good structure to aim at. Ligand-based design works by analogy instead. When the target structure is poor or unavailable, think of those disordered, hard-to-predict targets we flagged last episode, you design by resemblance to molecules already known to be active. The 2 are complementary, and the choice is dictated entirely by your structural information. Which is one more reason the structure prediction work in episode 7 quietly gates everything that happens here. Now the reality check, and it's the same bottleneck we keep hitting. Generation is cheap, validation is not. A model can propose thousands of novel molecules in a few hours. A wet lab can synthesize and test only tens of them per week. So generative design is inseparable from a scoring and triage layer. The model doesn't just have to invent good molecules, it has to rank its own output well enough that the handful you send to the bench are the right handful. A generator that produces brilliant molecules but can't tell which ones are brilliant is nearly useless, because you simply can't afford to make them all. And the existence proof for all of this is rentosertib, the drug will follow end to end in the final episode. It's an AI-designed small molecule, aimed at an AI-discovered target, TNIK, from episode 6, and it reached positive phase 2A data in humans. Hold on to that exact phrase, AI-designed and AI-discovered. This generative stage supplied the molecule, episode 6 supplied the target, and the fact that both halves were AI-driven is precisely what made it a first for the field. Generation built the needle, the next episodes are about making sure the needle actually works.